Guys, welcome back to my YouTube channel.
Biologically Frolic by Aishwarya. First of all, I want to wish you a very happy new year. May God give you everything that you want, maybe happiness, success, and traveling, everything. So, in today's session, we will be discussing about a very interesting topic called as a cell. A cell is called as a basic structural and functional unit of life. It means that we are all made up of cell. Not only us, it is a prokaryote, that is bacteria or some microorganisms. It can be animals, it can be reptiles, it can be amphibians. So whatever, how many species are available in today's world, they are all made up of cell. So in today's session, we will be discussing about a very interesting thing, that is history of cell, definition of cell, how many types of cells, and much more about it. So let's watch.
Guys, we'll learn about lots and lots of interesting things about cells. So, before we go to the next slide, there is something written in yellow color. Cells, the building blocks of life. Yes, cells are called as building blocks of life because we are made up of them. It is not only us, it is also microorganisms. From microorganisms to Pisces, amphibians, reptiles, aves, mammals, whatever the community we take, whatever the family we consider, everything is made up of cells, the building blocks of life. Let's go to the next session. So, before knowing what is actually cell, we should first know what is cell biology. So, cell biology is called as a branch of biology that deals with the structure, function, and behavior of the cell. So, it can be studying a structural aspects of the cell, or it can be a function and behavior of the cell. In what way it behaves, in what way it functions. So every cell has different functions. Cell present on the skin may work differently than the cell present in the liver. The cell present on the brain may work differently where the cell present in the intestine. Every cell has its own function, its own metabolism, or its own way of making a function. So, before going to actually cell biology encompasses both practical and eukaryotic cells, and that can be divided into many subtopics, which may include the study of cell membrane, cell communication, cell cycle, its biochemistry, and cell composition. So, you can see here that cell biology gives two types of cell to us. One is prokaryotic cell, another is eukaryotic cell. Obviously, we'll be learning about each and every type of cells in the upcoming slides. So, before going to actual concept of cell biology, we should know first its history or its historical aspects. So, let's have a look at the history of cell biology. So, when the invention of telescope took place by Galileo, it made us possible to observe the farther objects like stars, planets, etc. But the invention of microscope enabled us to even view the smaller objects which are not easily seen through our naked eyes. It was for the first time Robert Hooke in 1665 cells were observed by him. He remarked that it looked like a strangely similar cellula or the small rooms which monks inhabited. So, thus he gave the name as cells. So, if you just observe the image here, the cells were looking like a smaller room, small part of the rooms that he named as cells. If you observe the microscope also, it was not so advanced just like how we do in the present day. We have so many types of microscopes available with us. It's like compound microscope, then dissection microscope, electron microscope, phase contrast, fluorescent. We have so many types of microscopes available today to view the smaller objects. But at that time, microscopes were not so advanced to view the smaller objects, so it was not so easy to find out the microorganisms or the microbial world. Robert Hooke was somewhere unaware of his observation that he was not actually observing the living cell, but it was just an observation of dead cell wall of the plant cells. We call it as corks. So, this observation did not give any indication of the observation of any living cells or living things or living organelles like nucleus, mitochondria, etc. But then, for the first time, Antony van Leeuwenhoek, he witnessed a live cell under the microscope. In 1674, he described the algae Spirogyra, and there are some evidences that he has also seen so many species of bacteria under the microscope. He was a Dutch businessman and also a very good scientist in the golden age of Dutch science and technology. He was a largely self-taught man in science and he is commonly called as father of microbiology. Yes, if someone asks you being a microbiologist, if someone asks you the question that who is called as father of microbiology, it is yes, obviously Antony van Leeuwenhoek, who is called as father of microbiology. And the reason he was so curious to know about the microbes that you can see that during his life journey he has made so many types of microscope. But among them, an interesting fact about Antony van Leeuwenhoek was when he had during the course of his life journey he had invented so many types of microscopes. But among that, the pocket microscope was an interesting one. Whenever he used to go out for outings and roaming around the nature, he used to always carry his pocket microscope into his pocket. And whenever he used to find something interesting to observe, immediately he used to take out his microscope and he used to start observing the things. So that is the meaning that he was so curious about the microbial world and also about the microbiology. That made him the father of microbiology. Another interesting thing to share with you is about the cell story. Yes, the cell story, the theory, cell theory story is very interesting. The cell theory or cell doctrine states that all organisms are composed of similar units of organization called as cells. This concept was actually articulated in 1839 by Sheldon and Swan, and it has remained as the foundation of modern biology. So in 1838, what happened was this Schwann and Sheldon were enjoying after dinner coffee and they were talking about their researches. Then Schwann described plant cells with the nuclei, but Sheldon described the plant cells. While having a talk with each other, Sheldon described plant cells with the nuclei, but that thing stuck into the Swan's head, and he just observed that, or he just told him that there were similar kinds of cells that he also observed in animal tissues also. So these two scientists were completely in confusion, and they immediately rushed to Swan's lab to look at his slides. So, as we say, great idea starts from a great coffee. So, this idea hit their mind that there is some similarity between both plant and animal cell, and they came to the conclusion of the cell theory. Now, let's see what the cell theory tells. So, first conclusion, what it gives is the cell is the unit of structure, physiology, and organization of living things. So, first thing, the first conclusion, what they give is cell is the unit of the structure, physiology, and organization in the living things. The second conclusion, what they give is the cell retains a dual existence as a distinct entity and a building block in the construction of organism. So it means that cell constitute as the building block. They are the initial elements to build the whole organism. So if we are available today or we are existing today, if animals are existing today, it is obviously because of the cells. So if you just go deeper into the biology, you will observe that even we are made up of millions of cells. That was his second conclusion. And his third most unaccepted conclusion was cell forms by free cell formation, similar to the formation of crystals, that is spontaneous generation. So, as we know that spontaneous generation theory was completely disproved by other scientists, as the spontaneous generation theory tells you that we have been arising from some non-living things or just spontaneously from some aliens or something like that, which is completely wrong. As we know today, we have been existing from the pre existing life, right? So, I'll tell you the story of pre existing life also. The question hits our mind that how we have been formed from the pre existing life and from where pre existing life came from. Again, we come to the same question that from where the first life was arised. Well, obviously, try to give you the answer regarding this question, but we know today that the first two tenets are obviously correct, but the third one is clearly wrong. The correct interpretation of cell formation by division was finally promoted by other scientists and formally enunciated in Rudolf Virchow's powerful dictum. That is, it was one of his book actually, called as Omnis Cellula e Cellula. All cells are only arise from pre-existing cell. So hence here the modern cell theory was actually found. Now let's see that what modern theory tells us. First thing to conclude is all non-living things are made up of cells. So here you can observe the picture. It can be a tree, it can be a baby, it can be an elephant. We are all born from the single cell, just like the building blocks of life. So, just like blocks, they build up the whole statue or they build up the whole building, just like bricks build up the whole building. So, to build a whole new, brand new building, we require bricks, sands as a basic elements without which no building can be built. So, just like that, if we want to build an whole organism, it's not of course we have built it. Okay, so if one has to one has to born, the basic requirement is cells. That was the first conclusion. The second conclusion was the cell is structural and functional unit of all living things. So what we are today or what we are looking as today, it is because of the cells. Cells give us the structure. See, animals are completely different from us. Okay, fishes looks completely different from us, then amphibians looks completely different from us. It's not only about the animals, even we as a human, we look different from each other. You don't look like me and I don't look like you. It is because whole of our cell is unique and it is made up of different DNA structures, because of which we both different look different from each other. It is not only about the structure, and it is also about the functional unit of the living things. So functional unit means what? All the functions are carried out within the cells. Okay, it's just like mitochondria gives you the energy. Okay, ribosome gives you the protein. So just like whatever the function has to be taken place in our body will get initiated from the cells only. That was the second conclusion what modern cell theory gave us. The third conclusion is all cells comes from pre-existing cells by division. It means that spontaneous generation does not occur. So again question hits our mind that from where this pre-existing life came. So there are some scientists, they try to give us the answer regarding it. It was the Miller's experiment was so well known among it. So what he experimented as, he by using the heat shock treatments, he combined few gases which were he created such an environment which was created when the earth was about to form. So that time so many gases combined together, just like hydrogen, nitrogen, carbon, then so many things. So they combine together to form a DNA structure. So as the DNA evolved, it had a capacity to multiply itself or to divide. So DNA started dividing. So slowly protein coat came around it. So after that one, it started dividing again. So after some evolution, Pisces were formed, fishes were formed. So these fishes, after some evolution, they had a capacity to even come on the land. So that's how amphibians were formed. So amphibians, they evolved into reptiles. Reptiles evolved into aves. They were capable of flying, and finally aves evolved into mammals. So now what we are today, it is the result of so many eras of evolution that took place million years ago. So it is not that we are spontaneously born. Okay, it is not that we have come from some random non-living things or something like that. We have been born from our pre-existing generation only. So that's why we say that we were a kind of monkeys, apes in the earlier ages. But now, as the course of evolution progressed, we are as a modern human being as we are present now. This was the story of the pre-existing life. The cells contains hereditary information which is passed from cell to cell during the cell division. This was the fourth conclusion what modern cell theory gave us. So our cell contains hereditary information which is also called as DNA, okay, which you also call it as deoxyribonucleic acid, because all the information about us will be stored into the DNA, which is passed from one generation to another generation. Then the fifth conclusion was all cells are basically the same in chemical composition. So it can be a bacterial cell, it can be a reptile cell, it can be a mammal cell. All cells are same in their chemical composition. All cells contains the same type of chemical component. What is the difference is only about the DNA structure. Only the DNA differs from one organism to another organism, but except for that, all the chemicals are same from bacteria to us. Then the sixth conclusion was all energy flow, that is metabolism and biochemistry of life occurs within the cell. So energy will be created by mitochondria. It will be passed from one organelle to another organelle. So all these things will be going on inside the cells only. Okay, so as you know that cell, it is a most powerful thing that we have in our body, because of which we are surviving. Now let's have a look at ultrastructure of plant and animal cell. So to begin with, we'll start with ultrastructure of animal cell. So guys, you have lots and lots of good pictures that I have put it for you. You will be having so many good creative visualizations here, so that to make you understand the exact concept of how animal cell looks like. Let's get started. You can see the complex structure of animal cell here. I am not going to highlight each and every function here because we have a lot of things to highlight in the upcoming slides. I'll just mention that the organelles present into the animal cell. So this is what a typical animal cell looks like. So to begin with, we have nucleus into our cell, which is called as the controller of the cells, which controls the whole cell. Then next to that, you have nuclear pores on the top of cell nucleus, which controls the or which protects the nucleus from the upcoming hazards. You also have plasma membrane, which is the outermost layer of the cell, and this plasma membrane is actually semi-permeable. It will permit some solid and liquid materials in and out from the cell. Nucleolus is something which is inside the nucleus, and it has so many different functions. Then nuclear envelope, it is the outermost thing of the nucleus. It is again a protective layer. Chromatin, yes, chromatin is something important which contains our genetic material into it. Okay, and to protect this genetic material, all these things, this nuclear envelope, nuclear pores, so all these things are there. It is because of the chromatin or DNA or hereditary material which is present inside the nucleus. Then next outer attached to it we have this rough endoplasmic reticulum. Okay, it has some different functions, like it keeps the ribosome safe on top of it. So you can see the tiny spots on the endoplasmic reticulum. They are called as ribosomes. So ribosomes are called as protein factory of the cell which synthesizes proteins. Then you also have some smooth endoplasmic reticulum. Then some cells may have cilia, but and some may not. So not necessarily all the cells have cilia. Very few cells have them, and rest of them does not. Then yes, we have Golgi apparatus. Golgi apparatus is called as the postman of the cell, which carries all the materials from the cell and it gives to plasma membrane so that it will pass in and out from the cell. Then also we have microtubules. Obviously, I'll tell you the function of microtubules in the upcoming sessions. Then you have centrioles present. Then you have peroxisome present, lysosome present. Lysosome is called as suicide bags of the cells. Then you have microfilaments present, mitochondria, which is called as powerhouse of the cell. So you have all such organelles present into the cell. So once if you go and practice yourself with this structure, yes, you may be able to describe all parts of the cell for sure. So this was the complicated view of cell. If you want to view the simplified format of the cell structure, animal cell, it is like this. So it just contains all the parts, okay, like mitochondria, peroxisome, endoplasmic reticulum, ribosomes, vacuoles, and all these things. Okay, we'll begin with each and every organelle. Okay, I'll just highlight about all the organelles present into the cell. So let's begin with animal cell structure. So animal cells are generally smaller than plant cells. Okay. So another defining characteristics is that the animal cell has irregular shape. Why? Because of the absence of cell wall. So cell wall will be the rigid structure that gives shape to the plant cell. But here, as we have no cell wall in us, our cells won't be in a particular shape. Rather, they will be irregular. So this is due to the absence of cell wall. But animal cell shares other cellular organelles with plant cells, as both are evolved from the eukaryotic cells. So as I told you that we as animals and plants, we are all from the same genetics or same generations only. So most of the aspects, most of the organelles will be shared by animal and plant cells. So most of the organelles are same into the plant and animal cell except for few like chloroplast and cell wall. Let's come to the first outermost layer of the cell, that is cell membrane. A thin semi-permeable membrane layer of the protein and fat surrounds the cell. Its primary role is to protect the cell from its surrounding, and also it controls the entry and exit of nutrients and other microscopic entities into the cell. So as I mentioned earlier, this cell membrane is called as semi-permeable membrane because it does not allow everything to get inside the cell. So for example, there will be some toxic substance which are available outside the cell. So if it permits a toxic chemical into the cell, our cell won't be able to survive. So that's why it will be very specific in allowing the solid and fluid into the cell and even sending out of the cell. So this is something very important role carried out by cell membrane. Next, coming to nuclear membrane, it is a double membrane structure that surrounds the nucleus. It is also referred to as the nuclear envelope. So nuclear membrane is double layered because the whole cell depends upon the DNA only. So if DNA has to be well preserved, your nuclear membrane has to be very strong. So that's why it contains double layer in order to protect the nucleus and the DNA within it. So nucleus is called as the controller or the head of the cell. It is an organelle that contains several other suborganelles such as nucleolus, nucleosomes, chromatins, etc. It also contains DNA and other genetic material. So overall, this nucleus contains all the material that has to be passed out from one generation to another generation. So it has to be well protected, and also it controls the whole cells. Now, next coming to centrosome, it is a small organelle found near to the nucleus which has a thick center with radiating tubules. The centrosomes are where the microtubules are produced. Next coming to lysosome, these are called as suicidal bags of the cell. Why? Because they contain some digestive enzymes which when the cells will get old or when the cell ages, that time this lysosome will digest the whole cell and it renews the upcoming cell or it allows other cell to born. So that is how lysosome is called as suicidal bags. Next coming to cytoplasm, cytoplasm is a jelly-like substance which gives the fluidity or it protects the moisture level into the cell. So the substance found within the cell nucleus contained by the nuclear membrane is called as nucleoplasm. So if the fluid is present inside the cell, we call it as cytoplasm, and if it contains into the nucleus, we call it as nucleoplasm. So this is the liquid or this is the fluid that keeps the cell moist. Coming to Golgi apparatus, it is a flat, smooth layered, sac like organelle which is located near the nucleus and it is involved in manufacturing, storing, packing, and also transporting the particles throughout the cell. So, as I told you, this Golgi apparatus is called as postman of cell. Why? Because it does the work of taking the protein or energy or whatever the things it has to transfer from one place to another. It will take that and it will deliver it. So that's why it is called as postman of the cell. Coming to mitochondrion, it is called as powerhouse of the cell, as everyone of us know. These are spherical or rod-shaped organelles with a double membrane. They are the powerhouse of the cell as they play an important role in releasing energy. So next coming to ribosome, they are called as protein factories of the cell because most of the protein synthesis, not most, actually protein synthesis takes place by ribosomes. They are small organelles which is made up of RNA rich cytoplasmic granules and they are the sites of protein synthesis. If we talk about endoplasmic reticulum, this cellular organelle is composed of thin winding network of membranous sac originating from the nucleus. So it is a folded structure which surrounds the nucleus and which holds the ribosome for the protein synthesis. Coming to vacuole, vacuole is the membrane bound organelle which is present inside the cell. It involves in maintaining shape and storing water, food, waste, etc. If you talk about vacuole of plant and animal cell vacuole will be smaller whereas the plant vacuole will be larger in shape. Nucleopore. These are the tiny holes present on the nuclear membrane which are involved in the movement of nucleic acid and proteins within it. So nuclear pores are the tiny holes on the top of nuclear membrane which allows the protein or DNA material to go in and out from the cell. Yes, coming to ultrastructure of plant cells, I don't want to highlight the whole thing about plant cell because as I told you earlier, plant and animal cells shares the same organelles except for just few organelles like cell wall, chloroplast, graphite crystal, and large vacuole. So except for this, all the organelles are same for animal cells and plant cells also. So I'll be just highlighting very important plant cell organelles only. Among that, the first one is cell wall. So cell wall is a rigid layer which is composed of either cellulose, glycoproteins, lignin, pectin and hemicellulose. Again, it depends upon plant to plant that what it contains richly. Some plant may contain lignin as the rich material or some may contain pectin or some may contain hemicellulose and cellulose as the rich material. It is located outside the cell membrane and it comprises of proteins, polysaccharides and cellulose. So the primary function of the cell wall is to protect and provide structural support to the cell. The plant cell wall also involves in protecting the cell against the mechanical stress and to provide and form the structure to the cell. So it also filters the molecules passing in and out from the cell. So as I told you, cell wall does not only give the shape to the cell, it also does the work of protecting the cell from outer hazards. Because see, we as a human being can protect ourselves, but plants do not. They cannot move from one place to another place. So if any danger arrives, they have no chance moving from one place to another place. So rather they adapt themselves in such a way that if any hazard comes, they are able to protect themselves. So the formation of cell wall is guided by microtubules. It consists of three layers, namely primary, secondary and middle lamella. The primary cell wall is formed by cellulose laid down by enzymes. If we talk about cell membrane, it is same with animal cell, which is semi-permeable membrane, and it does the work of transportation of proteins and some enzymes in and out from the cell. Nucleus is same as that of animal cell. I don't want to highlight that much, but I will obviously talk about plastids. Plastids is something very important, which consists of six parts. One is inner membrane, intermediate membrane, outer membrane, stroma, thylakoid and lamella. So this plastid contains the very important thing, we call it as chloroplast, which is the site for photosynthesis. Okay, so plastids are actually of two types. One is leucoplast, so they are found in non-photosynthetic tissue of plants and they are used for the storage of protein, lipid and starch. And another one is chloroplast. So chloroplast is a green color, a green plastid which is responsible to cause photosynthesis. So the chlorophyll absorbs light energy from the sun and uses it to transform carbon dioxide and water into glucose. Central vacuole, as I told you that vacuole is this is it occupies 30% of the cell's volume into the mature plant cell, and tonoplast is the membrane that surrounds the central vacuole. The vital function of the central vacuole, apart from storage, is to sustain turgid pressure against the cell wall. The central vacuole consists of cell sap and it is also the mixture of salt, enzymes and other substances. Golgi apparatus, as I told you earlier, I am not going to mention that again here. Same with ribosome and mitochondria. I hope you are clear about what is cell theory, what are cells, what are the parts of the cells, and all about the history of cell biology, normal plant and animal cell, and what are the organelles into it. So I also highlighted the basic function of each and every organelle. In our next video, I'll be dealing with in detail the structure and function of each and every organelle that I described today. So let's see you in the next video. Don't miss it. You may find some important information into the next video. Thank you so much.